All right, so the next team we have here is BoutIQ Solutions. BoutIQ is solving one of the biggest bottlenecks in regenerative medicine and cellular agriculture, the slow, expensive trial and error process of media optimization. Alice is going to be the presenter. Over to you, team.
The human heart is extraordinary. Every day, this powerful organ beats 100,000 times, pumping over 7,000 liters of blood. It's the steady rhythm of the heart that fuels us, drives us, and keeps us alive. But during a heart attack, that steady rhythm is broken, blood flow is cut off, and heart tissue is permanently damaged. The heart can't heal itself, increasing the risk for heart failure, a condition expected to cost the Canadian healthcare system $19.5 billion by 2040. And the truth is, there is currently no cure for heart failure. But there is still hope, and it lies in cell therapy. Cell therapy offers a way to repair the damaged heart. Stem cells are grown into heart muscle cells using a nutrient-rich liquid called media, which consists of dozens of ingredients to support cell growth. These heart muscle cells are then injected back into the heart to replace lost cells. But here's the catch. Current engineered heart muscle cells are ineffective due to their functional immaturity. This renders them unable to integrate properly with the existing tissue, leading to problems like dangerous arrhythmias. It's biological challenges such as this, alongside commercial and strategic considerations that have led several key players in the industry to pause their cardiac cell therapy regenerative programs. But this leaves BoutIQ Solutions uniquely positioned to resolve a key technical limitation, as we know that maturation is highly influenced by media. Cell culture media is foundational to every cell therapy product, but conventional media design is simplistic and tests only a few factors at a time. It creates media that is generic, while we know that different cells have different needs, and this media also ends up being unable to support functional maturity or communication because it's so focused on cell survival. But the issue isn't the cells, it's the media that they're grown in, and that's a problem that BoutIQ chooses to fix. We've already proven that better media generates better cells. In a previous study using an older version of our algorithm technology, we delivered a cardiomyocyte maturation medium that outperforms leading R&D products. It was licensed from the University of Toronto to the Axel Biosciences and commercially launched in 2025. Now, we're going to take this a step further to generate cardiomyocytes for cardiac cell therapy. And to do that, we've created HeidiNew. At BoutIQ Solutions, our proprietary AI ML algorithm, HeidiNew, is designing media that supports cells better suited for cell therapy. So unlike these traditional methods, we screen and evaluate hundreds of formulations with the ability to test over 100 ingredients. We have cells grown in these different formulations, and real performance data is fed back into our self-correcting predictive model. The result? Unique, high-performing formulations, each with new IP discovered in fewer iterations and at a fraction of the cost. Current competitors use methods like design of experiment and simulations or other machine learning models, but these generate insufficient hits. On the other hand, we have data-hungry models that tend to perform better, but end up being prohibitively resource-intensive. With HeidiNew, we can efficiently test formulations tailored to custom problems, generating a large number of hits with fewer resources. Our solution is the most likely to identify top-performing media formulations that can support functionally mature cells in a viable setting. In 2025, Stem Cell Network funded a commercialization proposal with the O'Day and Simmons groups with BoutIQ Solutions. And this collaboration also includes leading biomedical and cardiac translational research groups, Radischi and Laflamme, and they're experts who have had success previously commercializing therapeutics. So our goal is to use HeidiNew to discover and optimize cardiac cell media and generate differentiated cardiac cells. We will then complete validation on animal models with the intention of creating effective cellular therapeutics. The total global cell therapy market is predicted to reach $31 billion by 2030, with $9.8 billion of that attributed to the cardiac cell therapy market. we plan to tackle 5% of that market share, and this market is currently untapped. BoutIQ Solutions is more than a one-trick pony. We already have media in the pipeline to tackle more than just cardiac cell therapy, including other markets like immunotherapy and cellular agriculture. Since Heidi New is a platform technology, we can use it to support media development in different customized problems. We've already had incredible traction. The Simmons and O'Day Labs raised $1.2 million in academic funding for early validation. Since then, the company has raised $700,000 in non-dilutive funding that will leverage to accelerate development of our proprietary platform. And speaking of proprietary, our algorithm, Heidi New, is patent protected. We filed a PCT jointly with the University of Toronto and started with national phase filings. And we're currently in negotiations to transfer that IP to the company, as well as make other IP trade secrets. So with our non-dilutive funding in place and IP consolidation underway, we're building on our prior success with the R&D media to advance cell therapy. So for that, we're raising capital. This round is sized at $2 million to deploy our AI-driven media optimization platform for cardiac cell therapy. Funding from Stem Cell Network will take us as far as media discovery and validation in small animal models, but we need this raise to build on that momentum and complete large animal preclinical testing of cardiac cells grown in our media formulations. Along the way, we'll also refine our AI model, positioning us for more IP and patent filings. And in doing so, we'll be ready for future strategic partnerships. Meet our team. Julia O'Day and Craig Simmons lead with decades of experience in biological search algorithms and cardiac tissue engineering. Neil Callahan bridges the clinic and lab as a physician scientist, and our scientific team, consisting of Doris, Hedda, and myself, bring the technical experience to drive discovery and validation. We're also backed by great business advisors, including external expertise from Panos Chrysanthopoulos from Weekend Dev and Tim Key, former director of business development at Notch Therapeutics and TIA. BoutIQ Solutions is delivering next-generation cardiac science at the bench, in the bottle, and at the bedside. With your support, we'll take the next step toward a clinical-grade solution that doesn't just support cells, it supports patients, turning the hope of cardiac cell therapy into a reality. Thank you.
Great. Thank you very much for that presentation. This team definitely fixes more than just broken hearts, so it's fantastic. We're just going to wait in the chat for any of the questions to come in, but maybe as we wait, we can start off by who are your competitors in this space?
Yeah, so we have a few competitors, Sartorius with Cellgetics and Nucleus Biologics in terms of like the media discovery. But then the main aspects that we do differently are that we're able to look at 100 ingredients. Well, they haven't said that this is one of the things they can do. And this is really important in media since we need to meet those complex needs of those cells. And also we're able to use customized problems and address them. Whereas if you have a really complex AI model that's not lightweight, then you would need a lot of that historical data and it would require too many resources to address those problems.
Great. And some of the questions in the chat, are there any pilots that are ongoing?
So I think our current main drive is with the stem cell network funding, where we're working on that on cell therapy viable media to grow cardiomyocytes. And this will kind of take us through small animal, large animal testing, and eventually we'll get to preclinical stage. Although we do have like the other two contracts with BioPalette, which is for lab-grown meat, and then we're kind of designing media for them. And then we also have this other T-cell media that we're kind of talking to companies to see if we can license that.